The overall goal of this procedure is the isolation and characterization by droplet digital PCR of tumor-derived circulating ribonucleic acid from whole blood samples. This method can help answer key questions in the oncology diagnostics field, such as how to detect tumor-derived gene fusion transcripts from circulating RNA in blood samples. The main advantage of this technique is that it uses highly optimized methods for the stabilization, extraction, and detection of RNA variants present at low frequency in blood samples. Blood-based testing is a complement to traditional tissue biopsies with the benefit of utilizing a minimally invasive technique that can provide rapid results. Although this method can provide insight into clinically significant mutations in NSCLC, it can also be applied to other systems where the accessible and rapid identification of circulating RNA molecules in whole blood is needed. Circulating RNA for the reverse transcription reaction is isolated from blood samples from donors as described in the text protocol. Convert the concentrated circulating RNA sample to cDNA using a commercially available reverse transcription reaction kit, including random primers. Include a no reverse transcriptase control sample and a no RNA control sample. When the reverse transcription reaction is complete, isolate cDNA from the reaction mix using a commercially available